I'm Kseniia. I'm fourth-year student in Zhou's lab. In our lab, we use deep learning to study regulatory signals encoded in the human genome. And my project is this Puffin model that we developed to study transcription initiation in a human genome. And now this paper is available on bioRxiv, but actually it will be published soon. It will be published in Science, hopefully next few weeks. We know that promoter is a very important part of the gene. We know that no gene can exist without a promoter. The genes could exist without a coding region, introns, exons, but it cannot exist without a promoter. I started studying genomics 4 years ago when I studied the grad school, and I was very surprised when I was reading about the promoters that the textbook talks a lot about how promoter is important, but then there is very little information. So what are the sequences that define the promoter? Most of textbooks mention data box, but no other motifs. And over last few decades, there was a lot of great research done on promoters. We know that there are several promoter elements, motifs that were identified across several species, but human promoters often don't have any of those motifs. We also know there are transcription factor binding motifs that appear near the promoter, but we don't know how all these sequences, all these motifs work together to define that transcription initiation starts here. And we also don't know what are the rules between those motifs, how they all work. And because in our lab we use deep learning and machine learning, we decided we can use machine learning. We can formulate this problem as a machine learning problem and study it. The transcription initiation in the human genome, it's not restricted just to one single base pair. It actually signals that spread over the window. And the signal is a transcription start site collected from many samples. So this noise, or like not noise, but this little peaks that we see in the signal, it's not a noise. It is actually reflecting the mechanism of transcription initiation. And we were thinking we can develop a model that takes the sequence and predict the base pair resolution strand-specific signal. And then if we train a model like this, we can use it to deconvolve the mechanism of transcription initiation that is encoded in a sequence. We used 5 transcription initiation maps, the maps with the highest coverage. They measured with 5 different techniques. The main difference between these 5 techniques that cage, phantom cage, encode cage, and encode rampage, they measure mature transcripts, and GROCAP measures transcription initiation and the nascent level. So we can use the model to study the mechanism of transcription initiation for mature transcripts, but also we can study transcription initiation at the nascent level. We developed a model that we call a deep learning inspired explainable sequence model, and we called it Puffin. The Puffin was designed to study only promoter activity. So it takes only local sequences to make the prediction, and it works as a sliding window prediction. Puffin is really good at predicting the shape of the signal because shape of the signal is encoded in the promoter sequences. However, the magnitude of the signal, it's also depends on distant regulatory elements, and Puffin doesn't use the distant sequences. So it's really good in predicting the shape, but it's not always perfectly predict the magnitude of the sequence. We first validated Puffin on the chromosome sequences that were held out from the training. So for the Puffin, those sequences are completely new. And we can see that correlation between Puffin prediction and observed values are higher than the correlation between the most correlated experimental techniques. So Puffin is it's a deep learning inspired, we call it deep learning inspired model, because we first had a deep learning model, Puffin-D, that we used to study transcription initiation. It also makes prediction, transcription initiation signal prediction from the sequence, but it is a black box model. It's not interpretable. And then using some ideas or knowledge that we found with a Puffin-D model, we designed Puffin. And this is the main advantage of Puffin model. It's not that it can predict the transcription initiation signal accurately, is that Puffin can explain why exactly this signal was predicted and what sequence patterns and rules in the genomic sequence define this shape of the signal. So now in the next few slides, I will talk more about the design of Puffin. So the core of Puffin are motif activation and the position-specific effect curve of every motif. So Puffin learns the motifs that are important for transcription initiation for the promoter activity, but then every motif can have a different effect on transcription initiation. And Puffin represents this effect with a position-specific effect curve that looks like this, with the motif is in the center, and the curve shows how motif affect transcription initiation depends on the position distance from the motif. For example, here, this motif will activate transcription initiation right upstream, and then it will also activate transcription initiation further upstream, but it suppresses transcription initiation right downstream. So this is how Puffin represents the effect of every individual motif. But another important question, how Puffin represent the effect of several motifs or interaction between the motif. So according to Puffin, there is no strong interaction between any two pairs of the motifs. So according to Puffin, any motifs can be combined in a promoter and start a promoter activity. And all effects of all promoters that can be additively combined to predict the transcription initiation signal. And it's important to say that they additively combine because our data is log transformed, but on the raw count data, this effect is multiplicative. And because this, we assume, according to Puffin, that all effects are additive, this makes the interpretation of the prediction much easier. And the last important part of Puffin design is that Puffin uses three groups of sequence patterns to explain transcription initiation. I so far talked about the motifs because they are the most intuitive, but also Puffin uses initiator sequence pattern and trinucleotide sequence pattern. The Puffin needs three types of sequence patterns to represent all kinds of sequence dependencies that are present in the genome. All of the sequence patterns, they follow the same rules that I discussed on the previous two slides, but they have different role in transcription initiation. For example, motifs, they are the most important. They contribute the most to the signal. Initiators, they are much shorter motifs, and the motif has a long effect range. Initiator sequence pattern has a motif range only within the initiator itself, and it does not contribute to transcription initiation as much as motif contribute, but initiators, they fine-tune the base pair resolution of the signal. And the last group of sequence patterns are trinucleotides. Those are just very short 3 base pair long sequences. We use them to represent the residual dependencies that we could not represent with motifs initiators. And it's very convenient that Puffin can describe the promoter activity in most of human promoters using only these 3 groups of sequence patterns. And now when I talked about the Puffin design, I finally can show how Puffin works. So Puffin is a convolutional model that has only 2 layers. And on the first layer, Puffin learns the motifs. it's important to mention that here I'm showing the motifs and their names, but we don't provide this information to Puffin when we start training. All these motifs, all sequence patterns, they are initialized randomly, and Puffin learns them through training. And only after training, I could assign the names. And again, here I'm talking about the motifs, but the same principles apply to initiators and trinucleotides. So the first step of computation that Puffin performs, it computes motif activation score. This computation is matching the learned motifs to the input genomic sequence by performing convolution between those kernels that represent the motifs and one hot encoded genomic sequence, and then applying the soft plus activation function. The motif activation score, it's similar to motif matching score, but motif activation don't have negative values because we apply a soft plus function. Here, for example, model found NFY motif, green CCAT, and Tata motif. And the plot shows the activation score of different motifs that were found in the whole 1000 base pair sequence. The second layer, now in the second layer, Puffin learns position-specific effect curves that we discussed earlier. So it's when motif is located in the center and what effect this motif has on transcription initiation. So Puffin applies convolution between motif activation score and learned motif effect. And the output is basically it's a weighted motif effect weighted by motif activation. Next, following the additive rule, the Puffin sums up all effect per base pair. And in the model, Puffin just sums up all effects of all motifs, all initiators, or trinucleotides. But on this plot, for convenience, we show that all motifs summed up separately, and all initiators and all trinucleotides summed up separately. And then here is the sum of all of the three group sequence pattern effects. And we can see here that indeed motif contributes the most to the signal. The initiator is just this wiggly line. It does not contribute that much, but it fine-tunes this base pair resolution that we see in the signal. And then trinucleotide captures some residual effect that we cannot capture with motifs and initiators. The last step is Puffin applying the log +1 transformation, and the output is the prediction. And here are the experimental values for this promoter. So this is the whole process. It's very convenient that Puffin performs only 2 steps of computation, and only these 2 steps are needed to make accurate prediction for most of human promoters. Another amazing thing about Puffin and its interpretability is that we can now compute, we can quantitatively estimate how every base pair near the promoter contributes to transcription initiation activity. And in our paper, in this presentation, we use sequence contribution scores. I mostly talk about 2 sequence contribution scores. One is base pair contribution score. So base pair contribution score shows how much every base pair contribute to transcription initiation signal at certain position or over a certain window. And another score is motif contribution score. It shows how specific motif how much it contributes to the signal. Again, we can choose any position or we can choose any window, and then we can compute how much motif contributes to activity in that window. And that score is important because often motif can appear near the promoter, but then it will not contribute to the promoter activity. And using Puffin, we can actually now differentiate which motif or which base pair contributes to this specific promoter and which does not contribute. And the last thing is that Puffin from scratch learned the motif that needed to for transcription initiation and the effect of every motif. So now we can have a look at what motifs Puffin learned. Puffin learned that only 10 motifs can explain transcription initiation in most of human promoters. This is very convenient because 10 is not that much and not that many. And all of these motifs, they actually known motifs that are associated with transcription initiation activity. Tata and Y1, NRF, ETS, NFY, so on, SP motif. The only motif that is new, in a sense, it's a motif that we call the lone initiator. But then, if we looked at the motif, we can find there's some promoter elements they matches this motif. So it's not matching any existing motif like as it is, but we can find certain parts of this motif that match existing promoter elements. So as I said, all motifs were new, but now for the first time we can show the position-specific effect that every motif has on transcription initiation, and we can show this using this effect curve that Puffin has learned. I think I haven't mentioned it, but Puffin learns the effect curve of the forward version of the motif, so the effect on the forward strand, but also Puffin learns the effect of every motif on the reverse strand. And this is possible because data that we used for the model training is strand specific. And on this plot, the motif is located in the center, and the solid line shows the effect of that motif has on the forward strand, and the dashed line shows the motif that effect that this motif has on the reverse strand. And based on whether these two effects on forward strand and on the reverse strand are symmetrical or not symmetrical, we can divide all motifs into two groups, into directional motifs and into bidirectional motifs. So directional motifs, they have strong effect on one strand, but a very different effect on the other strand. And examples of directional motifs are Tata motif. And as expected, it activates transcription 30 base pairs downstream of the motif. And then another example is Y1. And here is also that motif that we call lone initiator. And this is why we call it lone initiator, because it has a strong activation within the motif length. And there is another 6 motifs that have a symmetrical effect on both strands. So we call them bidirectional motifs. Because the model was trained with base pair resolution signal, we expect that these motifs, they actually represent the mechanism of transcription initiation. And one evidence of that is that NFY motif, it has this 10.5 base pair periodicity in the effect curve. And this is correspond to 10.5 base pair long helix curve. So we hope that in the future, scientists who study molecular biology of transcription initiation, that they can show that the mechanisms that they found will be consistent with these curves that the model learned. Because we wanted to make it easier for everyone to use Puffin, we developed a web server powered by Puffin, and it was developed by Jian Shi. He's a data scientist in our lab, and I can briefly show how it works. So if you go to this web page or use a QR code, this is a user-friendly website, and it's interactive. You have the sequence, and then you can see all steps of Puffin computation. This plot shows the motif activation score in that sequence, and also the sequence is colored by the motif, which motif was activated, like Tata here, or again NFY. Then you can see the motif effect, and you can see which motif contributes to the transcription initiation. Then the sum of every effect and prediction and the experimental values. You can you can input any coordinate that you want, or you can find the gene that you like, or you can replace the sequence with a completely new sequence. And the nice property of nice feature of this web server is that you can interactively edit the sequence right here. For example, we can delete data and we can see how signal changes. So it immediately runs the model, and you can see how prediction decreased a lot. Now there is no data activation, no data effect. Or then we can also input some motif. I have Y1 motif here, so we can put it somewhere, and now you can see Y1 activation, and you can see how signal changes. Yeah, and sorry, and you can replace this signal with completely new sequence, and you can see which motifs were identified in the sequence and what is Puffin prediction. And there is more features, like you can see the opposite strands, you can see other experimental technique prediction and other species. So I think that's everything. It's about I thought everything about the Puffin design. Next, I want to talk about additional experimental validation of Puffin that we have, and then how we used Puffin to answer or study some of fundamental questions of transcription initiation. But do you have any questions about Puffin model? Maybe I can answer them now.
Any questions? Please raise your hand. Yeah, Julian? Or yeah, Julian.
Yes, yeah. So what I don't get is how come the motifs are identifiable? It's always you can always sum two filters and get an equivalent one. So here it seems that each motif really clearly mapped to one transcription factor. How come?
So the motifs, it's like a filter in the convolutional layer. So we trained many Puffin models, and we found the model that performed the best, but also then we don't have too many motifs, or that the architecture is not too complicated. So we had to find the model that performs the best, but it's still interpretable. And then the model learned some motifs. Then when they matched, and like when we perform all the steps of computation, it gives an accurate prediction. So we didn't know if this motif that we find as a existing motif or as a completely new motif. It's only later we could match them to existing motifs. And also we trained the model several times. So we kept only motifs that we see every time they appear in the training. There are some motifs that sometimes appear in the training, like in top, let's say not top 10 motifs, but top 20 motifs. Some of those bottom motifs, sometimes they appear in the training, sometimes they don't appear in the training. Sometimes they seem like a redundant, like for example, Tata, but slightly different version of Tata. So we did not keep that motif. So we had to throw away some motifs, but make sure that we can still correctly predict the signal. I hope I answered your question.
Yes, you did.
Thank you.
And then following up immediately, you shown some that were bidirectional and others that were not. Does that relate to the fact that the motifs are palindromic or not?
So not all. It's interesting that not all of them they are palindromic. Not all of bidirectional motifs are palindromic. I think NRF1 is one that is palindromic, but for example, NFY is not palindromic. Or other motifs. And it's interesting that, for example, Tata is quite palindromic. At least it has this palindromic part, but it's very directional. Yeah.
Thanks.
Thank you. There's a
Irene,
question.
I think you had a question.
Yeah, so I'm sorry if I missed this, but I was wondering like what exactly the data set was you used to train your model. And do you think that if you, okay, great. And do you think that if you trained it, okay, so you used a bunch of different public data sets. Do you think, so I guess, how did you handle the data coming from like different tissues and cell types? And do you think if you had like trained separate models for like different tissues and cell types, you might have gotten additional motifs that are related to like cell type specific transcription initiation?
Yeah, thank you for your question. I actually didn't mention that. This all we combined all cell types here. So we have 5 different techniques, but we combined all tissues, all cell types to get the highest possible resolution. We did train Puffin on the cell-specific data too, not on all available tissues, but we chose 5 and 20 top tissues with the highest coverage, and we trained the Puffin model. Actually, this is the motif that Puffin identified in all tissues. The motif set was the same. The only difference we could see is that for this, when we plotted the effect curve for all, let's say, 20 tissues together, so you can see the overall shape is very similar, but the magnitude is higher. Like for some tissues, like the effect that, let's say, like ATS has is like it's higher effect, like the magnitude is higher. But overall, the effect curve shape is very similar, and also the set of the motifs that we found is very similar. And it's probably because I think that tissue specificity will be regulated a lot by the enhancer, so like distant regions, not necessarily in the promoter. So we have a one part of my presentation when we talk how composition of the promoter affects cell specificity, but it's all linked to activation or to regulation by distant regions that we did not use in Puffin. Thank you.
Great, thank you.
A question in the chat, should I read it?
Yeah, I can read it to you. Do you see different features between promoters of protein coding genes and non-coding genes? Asked by Nicolas.
Yeah, so we did not look separately. We trained again, I didn't mention that. I can now mention this, that we trained Puffin only on the promoter sequences, and we used top 40,000 promoters when the promoters sorted by the expression. So the top 40,000 strongest promoters in the human genome. We did not filter out coding or non-coding regions since we. Sorry, your question was if we see different. Oh, if we see different. Different features. Yeah, if talking about the motif compositions, we don't see that any like this one of the property of Puffin that there is no strong interaction between motifs, and we figured out that because when we looked which motif appear in the motif combinations appear in the promoter, we didn't find any motifs that appear together. So from this, I can say there's probably no, at least based on motif compositions, there is no difference between coding or non-coding gene, or at least there is no strong difference that we observed. Thank you for your question.
Okay, and then Kos is raising a hand.
Yeah, so I was just quickly wondering, you in your first motif layer, you chose 50, length 50 motifs, right? Or 51, yeah, sure. Yeah, so the motif layer specifically was 51. Is there a specific, to me at least, to an unobserved, it feels quite big. Is there a specific reason why you chose such a long one? I guess with your new like long initiator, you did find something, but you would expect it to be many complexes together, right? You're not going to expect one or even just a few proteins to be anything close to 50 base pairs in terms of proteins.
Yeah, so its motif is 51 base pair, and I think you're right. It's you can even here on my plots you can see that most of motifs they are much shorter than 50 base pairs. Most of them probably more like 20 base pairs, 25 base pairs, and So when we chose the size of all of the sequence pattern, including motifs, it's when we trained Puffin with different architecture. We tried shorter motifs or longer motifs, and the 51 that worked the best. And probably it is because of long initiator, because long initiator needs all this 51 base pair, and it appears in many promoters. So we needed it to get the model, and there is no downside of using longer motifs, since there is just nothing. When we match this motif to the sequence, this part that is there is like not important, it just does not interfere with model.
Yeah, of course. And I take it from that you also found that for the long initiator, the exact distances really mattered. So the distances between like the submotifs that you see in there have to be specifically that size?
It seems so. Like the model did not, for some reason, model did not learn any of these motifs as a separate motif. It learned it as the one motif. So I guess this is because it's like the position of these motifs inside the initiator probably matters a lot. That's why model kept it together. Or it's also because I guess they probably appear together also. And yeah. Thank you.
Okay, I think we can move on to the presentation, and then maybe there's more questions at the end.
Okay. In the paper, we have used several already experimental data sets that were already published. So we wanted additional data sets because I mentioned before we validated Puffin on the chromosomes that were held out from the training, but we also wanted not just measure Puffin accuracy, but also find evidence for the position-specific effect curves that Puffin learned. And we found several data sets that we could use to do that. And here I'm showing just one example. In this study by Andrew Oldfield, in the study, the authors performed knockdown of NFI transcription factor. So they knock down the transcription factor and they measure transcription initiation signal, which base pair resolution transcription initiation signal before knockdown and after the knockdown. And we can actually replicate this experiment in silico because we can just turn off the activation of NFY motif. And we assume this is similar to knocking down the NFY motif. So we turn off the activation and then we make the prediction before this in silico knockdown and after in silico knockdown. So the first plot shows the activation of NFI in which places. So this is a plot. Forgot the name. Matrix plot, cluster plot, showing all 2,000 promoters. And here this is the activation of NFI in every promoter, and then we do the in silico knockout. And we take the difference between predicted signal after the knockout and before the knockout. And here we see that there was signal. Initially, the signal was in the center. Now it is shifted upstream. And this is the experimental signal measured with start-seq with the knockdown of NFI protein. And we observe that our prediction is consistent with experiment. Experiment also shows that there was signal in the center and then it's shifted upstream. So we had several experimental data sets that we used, but the problem with most of them, they are not measuring exactly what Puffin is predicting. For example, here, this is a knockdown, and so it's not like the genome was edited, it's just the protein amount was decreased. So it's not exactly the same thing as what Puffin is doing. And also this experiment was conducted in mouse. And the similar issues we had with other data sets. Sometimes they're not in human cell lines, and sometimes, like often, they don't measure base pair resolution transcription initiation. They measure just the expression value. And we still can use this data set, but it's not exactly what Puffin is doing. That's why we wanted to develop a new assay that we can use to specifically validate Puffin effect curves. And this is an assay that was developed by Dudnyk. He's a postdoc. He's a joint postdoc in our lab and in Jian Shu's lab and St. Jude Children's Hospital in Memphis. And he developed this assay. He called it CRISPR-Cap. So here he used CRISPR-Cas9 editing to delete the motif. And then he measured the transcription initiation signal before deletion and after the deletion. So it's just deleting one of motifs that Puffin identified and deleting it in a human genome. The limitation that this assay has is that we can only delete the motifs that is downstream of transcription start site. And there is only 2 motifs that are downstream, that are located downstream in human genome. It's YY1 and the lone initiator. So we deleted YY1, we deleted lone initiator, and we also did the YY1 shift because we can delete some region between TSS and YY1. So it causes a shift, and we can see that can we accurately predict the effect of motif deletion or the motif shift. And on this plot, so here I show the experimental values for the wild type promoter, for the mutant for promoter after motif was deleted, and here is a prediction. And on this plot, we can we should look at the shape. So can we accurately predict the shape that was measured? And overall, we had 16. We edited 16 promoters. For 11 promoters, we had high enough coverage to look at the results. And for also 11 promoters, the prediction is consistent with what experiments show. Like if we look at the shape, how it changes, it's all in agreement with Puffin prediction. So using Puffin, we now can compute how every base pair contributes to transcription initiation. We also now know what sequences define the promoter in human genome and how those sequence patterns interact with each other, how they form the signal. But there are several fundamental questions about transcription initiation that are not completely clear, and there was a lot of research done on these questions, but we wanted to see if we can also contribute, if we can use Puffin to explain or understand some of those problems. For example, is the composition of the promoter, does it affect the function of the promoter, and whether Puffin can explain the strand specificity of promoters, and whether those rules that we found in human genome, are they conserved across many species. So one of the questions is, can we use Puffin to explain the strand specificity of the promoter? When we look at the transcription initiation signal, we always see where promoter we see the forward signal on the forward strand, but we also saw almost always we see also signal on the reverse strand near the promoter. And the amount of the signal on the reverse strand, it depends on the technique that we use. For those techniques that use total mature transcript, the signal is small, but for NASA and the NASA level, these 2 peaks are comparable in the intensity. And we want to see if Puffin can explain this and the difference between these techniques. So Puffin can qualitatively explain the directionality of the promoter. As I mentioned, there are directional motifs and bidirectional motifs. So these bidirectional motifs can create transcription initiation signal on both strands, but directional those motifs that will explain why the signal on the forward strand is much stronger than the other strand. But also there is one special motif, UR-SRNP. So this is actually corresponds to 5' splice site. And we see this motif only when Puffin is trained on the mature transcripts. For those 2 techniques that measure nascent transcript, transcripts 5' beside this UR7P, it does not appear. And this is what explains this difference between these two techniques. Then there is another question that was not completely clear. So we do observe there is signal on the forward strand and on the reverse strand, but do they share the same promoter, or is it just two promoters that appear near each other? And using Puffin, we can quantitatively assess the contribution that the sequence has to the transcription initiation on one strand and the transcription initiation on the other strand. So how we can do this? We can predict the strand-specific signal, and now we can plot the contribution of every base pair to the forward signal on the forward strand and contribution to the signal on the reverse strand. And then we also color the signal by the motifs that matched at this position. And here we can quantitatively see that the same position contributes to transcription initiation activation on the forward strand and on the reverse strand, and those are the same, also the same motifs. Next, we wanted to see if the composition of the promoter has effect on its function. What we can do, I mentioned the base pair contribution score. But here we can use a motif contribution score. So this example how motif contribution score is computed. We have sequence. We show this all effects that motifs have on this signal in the center. So we now can compute how much every motif contributes to the transcription initiation signal. And we can do it for all promoters. And here, for example, we have top 100 promoters, and they are clustered, but their motif contribution to the activity of that promoter. And then we divided all promoters into several groups. So for every motif, we sorted promoters by the contribution from this motif to the signal, and we chose top 1,000 promoters for every motif. So we had 10 groups of promoters with 1,000 promoters in every group, and every group has a high contribution from corresponding motif. And then we want to see if we see any difference in the functions of that promoters in every group. And what we notice that there is the composition of the promoter is linked to the expression selectivity of the gene. We computed this dispersion index. This index, it's a variation of promoter expression across tissues and the mean expression of this promoter. And we plotted it. So on this scatter plot, you can see every dot is the promoter, and it's colored by the contribution from the corresponding motif. So for example, here the dark dots, the motifs, the promoters that did not have contribution from Tata, but bright yellow dot that they have a high contribution from Tata motif. And so on the y-axis, it's this dispersion index. On the x-axis, it's a mean expression. So those promoters that are tissue specific would be on the top, that are not that ubiquitous would be on the bottom. And when we plotted this for all motifs, we can see that Tata, there's two very distinct motifs, Tata and YY1. Tata promoters, they are highly, they very tissue specific, and they highly expressed, so they're located here. But YY1 promoters are located here, so they also pretty strong, but they are ubiquitously expressed across tissues. And all other motifs or other motifs have effects somewhere in between, so they can be sorted from Y1 ETS that are more ubiquitous to SP and Tata that are the most tissue specific. And then we have UR-SRNP, which will be a negative control because its effect is post-transcription initiation effect. So it should not have effect on tissue specificity of the promoter. And that's what we can see, that dots are everywhere. So we did observe that there is a link between motif composition and tissue specificity, but we wanted also to explain this link. And we wanted to see if we can study this if we take the promoter and insert in different sequence context in different locations in the genome. So we chose those 40,000 promoter sequences and we inserted them in many genomic locations. Some of those locations we had higher transcription initiation activity. Some of them has no transcription initiation activity. And we need to make the prediction of this activity. And this is where we cannot use Puffin because it uses only local sequence context. But we have Puffin-T model that takes a long sequence and makes a same base pair resolution prediction. So we did that, and we observed that similarly to what we see across tissues, we also see that Tata promoters, they are more selective. They only expressed in few locations, and Y1 promoters, they expressed in many locations. And another thing we observed that it's probably connected to how the promoter responds to activation of distant regulatory elements. Here, we plotted the response curve of every group of promoters. And the y-axis, this is the expression of the promoter in that location, genomic location where we inserted it. And here, y-axis shows the rank of this location activity. So this is mean predicted expression of that location across all inserted promoters. So these locations in the genomes that already had strong transcription initiation activity, they will be on the left, and those that weak will be on the right. And we can see that data is, when it's inserted in a strong location, its activity grows even more, but then it's very responsive. But Y1 is the opposite. It's not that responsive, and all other motifs are in between. And if we look at the order, how this motifs is ordered, it's similar to what we see with cell specificity or selectivity across genomic location. The responsiveness, they ordered the same way, and Tata is the most selective and most responsive, and Y1 is less selective and least responsive. So this is a link we found with between motif composition, promoter composition, and its function. And the last thing we studied is if we can see whether those rules of transcription initiation that we found in the human promoters are they conserved across other species. The problem is that there is not that many data, base pair resolution data available for other species. The only species when there is a lot of data, it's a mouse. So we trained Puffin model on mouse data, and then we used mouse model to predict transcription initiation signal in mouse genome. And then we also applied human model on the mouse genome, and we predicted the mouse transcription initiation signal. And we can see that this all was evaluated on the holdout chromosomes. And you can see that the mouse prediction on mouse model is very similar to human prediction and human model. And overall performance of these two models is also very similar. The correlation between predictions is 0.996. So it seems like, so we can just use human model on mouse data, which seems that shows that these transcription initiation rules that we found in the human, they probably conserved a lot in the mouse too. And because we assume that we can use human model to predict transcription initiation signal in mouse, we can also then use this human model to study transcription initiation or to predict transcription initiation in those species that are between human and mouse. We used 241 mammalian species from Zhou project. We use aligned promoter sequences. And we wanted to see how the motifs that Puffin identified, how they conserved across all these 240 species. So here we plotted the conservation score for every motif, and this line is it's actually all 240 conservations motif activation plotted. And so we can see that the motif is conserved at the position that is the most optimal for transcription initiation. For example, data is conserved 30 base pairs upstream of the TSS, and Y1 is conserved downstream of the TSS, because Y1 activates transcription right upstream of the motif. And even this periodicity of NFY is also conserved. And what's amazing that this curve, this conservation of this position, it's also the same as very resembles a lot the effect curves that Puffin learned, like the NFY, Y1 data, and so on. So this is another indirect validation of the position-specific effect curves learned by Puffin. So this is the end of my presentation. So we used Puffin to create unified view of transcription initiation. We can explain the promoter activity with Puffin in the most of human promoters. We can now identify how exactly every base pair contributes to the promoter activity, and we found the link between the motif composition of the promoter and the gene expression selectivity, and we also showed that the promoter activity on both strands is likely shares the sequence basis. And we also showed the rules of transcription initiation are conserved across mammalian species. We built a web server that now everyone can use to study their favorite gene. And we also think that this approach that we use for Puffin, so we have first developed a deep learning model, and we studied the transcription initiation with deep learning model, and then we use some findings or ideas from deep learning model, and we built a new model that is simple and explainable, but also make the accurate prediction. So we think that this approach can be also used to study other regulatory signals in the genome. And I want to thank my lab. We now have a lot of people in our lab. When I started, we had only 3 people, and now it's a lot of people. And especially I want to thank my PI, Jian, Zhou, and everyone who worked on this papers, Chen Lai and Don Hong, and our collaborators in Jian Shu's lab. Thank you for listening to my presentation.